So hello, I'm Jan Krause. I'm a state archivist in Canton de Vaud, next to Geneva.
Good afternoon. My name is Hugues Cazeaux. I'm working with Pierre-yves at the University of Geneva for preserving research data at the universities.
Okay, thank you. So, good afternoon. I'm going to speak about this OAIS-compliant digital archiving using DNA. And we start with setting is what's the problem. Probably you know about this problem stated by Rosenthal in 2010. This is a requirement of archiving 1 petabyte for 100 years. And this is quite challenging, as you can see in this slide. And according to the CERN study, we could actually evaluate what is the half-life of bits, and it was found to be 10 to the power 8. But to guarantee this survival of 1 petabyte over 100 years, you need, there is a gap in technology of 10 to the power 10. So maybe DNA is a solution. Why? Because DNA half-life can be very long. It can be 1,000 years, even more or less. It depends how you conserve it. But also, it's very easy to make copies. You probably heard about PCR at this COVID time. So it's very easy to make copies. It's just thermal cycles. You can get 10,000 copies in a few hours. Also, we know the statistic of error of DNA. It's quite well studied. And so after 100 years, the problem is you still have 93% of your DNA. So that means you have 100% of information still there. For storage preservation, so depending how you preserve DNA, can be the half-life can be shorter or longer. But the nice thing I want to show you here is that the ideal temperature to keep DNA is about 15 degrees, depending the technology. But the good news is that the new buildings, the modern buildings, use passive energy precisely in this range of temperature. So it would mean that it would cost nothing to keep information, no CO2, nothing special. Just to remind you about the density comparison about DNA. So here, for 40 zettabyte, you would need 42 billion of USB sticks. You would need 2.6 billion of hard drives, or 227 million of tapes. And this is not a dust there, but it's just a single gram of DNA to store everything.
Thank you. So I will tell you a bit about what we are doing more concretely. So the idea to manage the DNA storage is to do this life cycle, which is encoding data to logically to DNA, and then writing it, it's DNA synthesis, then storing it, so using a method of preservation, then reading it back, it's sequencing, and then decoding the DNA back into data. So obviously I won't get into details because we have only 12 minutes, but just a few words about encoding and decoding. So what we started to do is to work on strong scientific basis, so it's robust, and we use the work of a lab at ETH Zurich of Professor Krause and also other researchers. The idea is also that the code that we wrote to do this must be sustainable, because we need the code to be able to read back the data in the DNA. To this, the library that we have developed is relatively simple, so it's sustainable. It's one language, it's pure Python, no dependencies. It's based on algorithms that have made the proof, so it's Reed Solomon, is used since over 40 years. It's in CDs, DVDs, Blu-rays, and other protocols. and all the code is fully documented. There is a basic configuration allowing to store AIP from small size, a few kilobytes, to big ones, so gigabytes, without having to change the configuration. And it's very short, the code, so you can print it on a few sheets of paper, like 20, and on long-term preservation paper, and use it to document the DNA storage so it can be read in the future if we lose the code itself in the computers. So, and it's open source, you can find it on GitHub. Voilà. So just to explain a bit more how it works, so imagine it's an AIP, okay, here it's text, but it could be anything that is binary. It's just to illustrate, it could be bits. In fact, it works with bits. So the idea is to reorganize the data into a table. So here it's vertically, and it's put vertically into columns. And then we use this error correcting code, read Solomon, that doesn't change the data, but adds code so we can restore a column if it gets damaged. And then we number the columns, 1, 2, 3. Right? Then we apply the Reed-Solomon algorithm in the other direction, so we can restore rows if a row gets damaged. And then we convert it to DNA using a simple code, like the bit 00 becomes A and so on. And then voila, it's finished. And with the default parameters, we can correct up to 32 mutations or errors in one fragment. With the default parameters, fragment can go up to 2,000 nucleotides, and it's set so we can lose up to 20% of the fragments, of the segments, but it's just a parameter. So we could do more. These parameters are set that way because of the scientific studies that suggest that this is the right thing to do, so we can read the DNA over time. Thank you.
So me, I will introduce the technology. So I will be, in this project, we plan to implement what the proof of concept will prove, in fact, in the DLCM technology, which is based on the OAIS model. So I won't go in the detail of this architecture. I already introduced this technology during the iPres in Amsterdam in 2019. So if you look at in the archive of the iPres, you should find the poster session corresponding to this technology. So I'm more focused on the storage. So this technology will be the custodian of the OAIS standard, in fact, because by design, this technology respects the OAIS model with all the different modules, with the ingestion, the archival storage, and the dissemination. And in this technology, also by design, we also supported two kinds of storage. So the file storage and the object storage. So in the code, in fact, we already think about to open, in fact, the implementation to other technology. The objective is to in fact, implement what the proof of concept demonstrates in DNA. So that is the architecture point of view. From more detail in the implementation point of view, we just in this table show you the different functions that the DNA has to support. So for storing an AIP, you have to generate the DNA, to synthesize it, and to store it physically. So it could be, you could have parallel with physical archive. And also you need also to support also what OAIS expect from an archive is to have an auto-sufficient documentation. So the idea also is to be able to retrieve, in fact, metadata in the DNA. So by thinking of the software implementation, we are able also to define some requirement in the DNA implementation. I'm a software engineer, so but I'm not a biologist. But with this reflection, we are able to think of new requirements for the DNA storage.
Voilà.
Voilà.
Okay, so the next step after integrating into this OAS architecture is we would like, because archivists are not biologists, so we want to offer them the possibility to read information without having any knowledge in biology. So we want to industrialize the reading process end to end. This is shown schematically here, the different steps you have to do, and a machine is going to do it actually. So the idea is to use the macronin lab concept, which has been developed in Switzerland for fabricating watches. We want to apply it to DNA. So this is small cubes where you can make micro machinery inside, and this is going to be the solution for automatizing the process. So thank you for your attention, and if you are interested in collaborating with us, we are looking for partners for the next Pathfinder Challenge, European project, on DNA-based digital data storage. So just contact us if you want. Thank you.
Thank you so much, and also being very good on time. So, is there any questions? Yes, there's one there. We just need, if you state where you come from and your name, and we need a mic down here. He's coming now. Yeah.
Hello, my name is Daniel Salverovich. I'm from National Library of Norway, and I just had a question of how do you think about using that in the future as an active storage, or would it be more of a second or third copy that you keep just in case the more readable format like a hard drive explode or something?
As an archivist.
Yeah, so first of all, it's a question of future, right? Because now it's too expensive. But the price of storage is increasing, and the price of DNA synthesis, which is the most expensive step of the process, is decreasing. So that's the so that's what will, I guess, condition how we can use it. And then about your question more specifically, I guess, we have a lot of archived data that is not used, or used just a little bit. Like a small percentage of the data is used a lot, and a big percentage of the data is not used much. So I guess this would be very interesting for the data that is not used a lot, because it takes time to access. And so it would be maybe interesting to put it in DNA, and the rest to have live copies. I'm not sure I answered everything, but...
Yeah, that was it. Thank you very much.
There's a question. Oh, is there anyone left? No, okay.
Alexander Roberts, Swansea University. Really interested in the persistent URI that you alluded to as DNA : / under development. And just a question for, around retrieval. What's happening in that space, if you know, please?
It's under studies today. But the idea is to define, in fact, a kind of URI for DNA, in fact. That is the spirit of my slide, in fact. Because for implementation point of view, we need to be able to have a notation, in fact, to identify, in fact, the IP. So it's just a replication of what we've done with object storage or file system, but that is the idea. But today we don't know, that is the plan. We have to think about it and to study, to work on that, to see if it is the right definition, or to find something new.
What's the time? We'll take another one. A very small question. Is there one from there? Yeah.
Jonathan Isip, University of the Philippines. Just a quick follow-up to the first question. Do you have write speeds and read speeds for the encode and decode?
Read speed and write speed.
Write speed is very slow. No, because synthesis is still in development, I would say, as a technology. But so for us, it's why we don't want to automatize synthesis writing, because we have to use companies, specialists for that. Reading, that's a question, because if we develop this automaton, we can also parallelize the process. So if it is too slow, we can put the different the same reading process in parallel to accelerate. But no, we don't have numbers, really. It depends on the technology. If you use Illumina or NanoPore, for instance, it will be different. And we are too early in this prototype for now to give you a number. But what is sure is that you can accelerate it in the future, for sure. Yeah.
Thank you so much. And just so you can ask afterwards, there will be time where you can, if there were any more questions, but we don't have time. But thank you so much.